Hi, this is Alexa Tatarian, Project Coordinator at Nelson Laboratories. Thank you for joining us today to view this webinar on cleaning validations for newly manufactured medical devices and single-use implants. Again, my name is Alexa, and I'm a Project Coordinator, and I specialize in consulting on these types of cleaning validations. And today we're going to talk about some tips and tools that you can use on designing your validation. These validations also will look at residues remaining from the cleaning and manufacturing processes. To begin, to ask, what does it mean to be clean is a question that's often asked with these cleaning validations and what we're talking about today. And I have a picture of a carrot up here, and you can see it's dirty, and you can see visible that's dirt that's left behind on this carrot. And kind of the process on how to get this carrot cleaned, ready to consume, it goes through a cleaning process. And along with the dirt that you can see with your visible eye, there could be contaminants left behind, such as pesticides or other chemicals that maybe you can't see. And so, kind of like with medical devices, these cleaning validations are really important to remove these residues before they see the patient. And an important note to remember with these cleaning validations and why they're so important is that just because something is sterile and goes through a sterilization process doesn't mean that it's necessarily clean. And so these cleaning validations are very important to assess the cleanliness of these devices before they're implanted into patients. So what is a cleaning validation? A cleaning validation is documented evidence that the cleaning process is effective and consistent at removing residues and contaminants from the manufacturing and cleaning process. And there are three real important points to note with this definition. The first being that this is documented data. If an auditor were to come to your facility and ask if you were producing and manufacturing clean devices and you say yes, how can you back up that claim? And what data do you have on file to show that auditor? You also want to make sure that the cleaning process is effective. Can it remove those residues from the devices? And thirdly, is it consistent? Can you consistently get the same level of cleanliness lot to lot, and is your process reproducible? Along with this definition, there are some goals and purposes of a cleaning validation that we also have listed here that are important to consider in the cleaning validation. You know, are you removing those manufacturing residues, the cleaning residues? Are you producing a biocompatible device? Also, you want to reduce the amount of microbiological contamination present, such as bioburden or endotoxin. And finally, are you just trying to improve the overall cleaning process? A lot of people may want to implement a change or a new step in the process, and these cleaning validations can help you decide after you've sent your benchmark data if those changes are beneficial to implement or not. So why is it important to validate? First, it's important to know that this is a requirement. It's a GMP or good manufacturing practice requirement that manufacturers shall have procedures in place for the use and removal of such manufacturing material to ensure that it's limited to an amount that does not adversely affect the device's quality. And so it's important to note that this is actually a requirement. A lot of people call and ask, you know, why do I have to do this testing? And, you know, this is regulated, and there's a regulation on these validations. Along with that regulation, it doesn't talk about what types of residue that you should be concerned with or limit values or acceptance criteria, which makes it difficult for the manufacturer. But hopefully this presentation will give you some tools and tips to use on how to design this cleaning validation. There are also other benefits to performing a cleaning validation, one being to satisfy regulatory authorities. These validations are really a hot topic in the industry right now, and we've seen increased awareness from regulatory authorities such as the FDA, where they're starting to ask more manufacturers for this validation data, and they want to see it on file. And so we've seen definitely an increased awareness recently of people having to do these validations. And most importantly, a benefit of these cleaning validations is for patient safety. There could be residues left behind on these devices that could be cytotoxic, which could be very harmful for patients if implanted. Also, with regards to patient safety, the effect to the function of the device should also be examined as part of the cleaning validation. For example, there was a large lawsuit and recall about 10 years ago where there was so much oil that was remaining on these implants, it was a hip implant, that the oil was acting as a barrier between the implant and the bone, and the patient's bone could not grow and integrate into the implant because there was so much oil residue left behind. And interestingly enough, that oil and those devices were showing up as not having a cytotoxic effect. And so again, kind of going along with that concept of just because something is sterile and non-cytotoxic does not mean it's clean. And so the effect to the function of the device should also be examined with these cleaning validations. There's not too much guidance in the industry on these cleaning validations as a whole. However, there is one document in place, ASTM F2459, which was published in January 2011, which discusses these cleaning validations as a whole. And this standard outlines test options that you can use to show device cleanliness. And this standard pairs standard microbiological test methods, such as bioburden and endotoxin, with analytical test methods to quantify the amount of residues that are left behind. And together, these tests really show a comprehensive view of the cleanliness of the device. It's important to note with this standard that we are really just focusing on surface residue contamination here and not any leachables. And so that's an important note with these cleaning validations. There are also two working documents in place that we wanted to mention that the ASTM has out that have to do with these cleaning validations as well. The first is a working document for designing devices for cleanability. And this is a trend we're seeing where people and manufacturers are starting to think of the cleaning validation up front when they're designing the device as well as the overall process. For example, when designing a device, maybe thinking of the questions, how easy is this to clean? And does this hinge maybe, would this hold on to more residue? Is this blind hole maybe going to cause problems in the end? Same with designing the process itself. Maybe one detergent is more sudsier in the process and it would be more difficult to remove. Can you get away with another detergent perhaps? And so this concept of designing for cleanability is something that's being thought of up front before the cleaning validation to try and reduce as much risk as possible. The second working document that we wanted to mention is there is a working document in place for establishing limit values on these single-use implants. And so that's a document that's going to be in hot demand when it comes out, and more guidance on how to justify these residue levels, as there is no set acceptance criteria in the industry currently. And so also to note with this working document that in the last ASTM conference that we attended, we do not think that this working document will actually set a limit and actual numbers, but it'll just be more guidance on how to go about setting those limits. And so it will still be up to the manufacturer, in our opinion, to justify that overall test plan and clean devices. But to mention these two working documents, we're watching those to come out in the future. So when to validate the cleaning validation and the cleaning process? The initial validation should be performed when there's no previous data on record for this process. Also, the validation should be re-examined if there was a change made to the validation or cleaning process, such as a change in the step in the process. Maybe an extra step was added, a change in material, and material to either the device or a new component in the process as well, or a change in location. Even if all the steps in the process remain the same and you just move locations or maybe add a new vendor, we recommend revalidating or retesting just to make sure that there are no other contaminants that were introduced with this change that could be a problem for your device. And an interesting example with this, we were recently working with a client who made a change in their process where they added an IPA rinse in their cleaning process to their device. And their device had a tubing component. And with this IPA rinse, it was getting, this customer was doing routine monitoring with TOC, which is one of the test options that we'll go over later in this presentation. And they were seeing a spike in their TOC data. And we were talking with them saying, you know, have you made any changes to your process? You know, where could this spike be coming from? And they were telling us about the IPA rinse, and they were able to conclude that in their process, the IPA was not being allowed to evaporate in that tube, and so there were residues being left behind. And so because they saw a spike from this, you know, continual monitoring after a change was made, they were able to go back to their process. They added a water rinse after the IPA rinse and were able to flush out those residues left behind, and they were able to get their levels back down to an acceptable level of cleanliness. And so it's really important to note if you've made a change in your process to kind of reevaluate things to make sure that there are no other contaminants that could be introduced that could be harmful on your device. So this is the validation plan and kind of the outline for what we're going to talk about for the rest of the presentation. First, it's very important, if possible, to determine the potential contaminants that come in contact with your device. And this will be important for step 2, choosing the appropriate test method based on those potential contaminants. And that's step 3, justifying those residue levels. And we'll go over some tips and what we've seen from other companies and of what have had success to give you some guidance on how to justify those residue levels, again, since there's no acceptance criteria for these validations as a whole. In determining the potential contaminants, you want to look at the manufacturing and cleaning process and really anything that comes in contact with your device that could be possibly left behind. So from the manufacturing process, maybe you have oils, lubricants, polishing compounds, things of that nature that come in your device that could possibly be left behind. And also residues from the cleaning process, detergents, alcohol rinses, you know, what's coming in contact with the device that could be left behind. And then once we get that list of potential contaminants, we divide them into groups based on their solubility in water. And so these contaminants are either going to be water soluble, non-water soluble, or insoluble debris. And breaking these down into these groups is what is going to help us choose the test methods that are most appropriate to quantify those residues. And so getting this list is very important. Sometimes this can be a very lengthy list. Sometimes devices come in contact with many, many different potential contaminants. And often for the validation, our clients will maybe pick their worst case contaminants and focus on those for the validation. Also, it's common that our clients do not know either what the potential contaminants can be. And in that case, we recommend trying to examine as many of the unknowns as possible to try and cover your bases, which we can also address later in this presentation. So in choosing a test method, there are 4 options available to quantify those surface residues. And then based on those solubility of the potential contaminants, one or a combination of these tests would be beneficial. And all of these tests perform a device extraction, and then that extract is examined to quantify the surface residues. And so we're going to go in depth now a little bit more about each of these test methods and what they can do. So the first test method we're going to talk about is the quantification of extractable residue by gravimetric analysis. And this test method has its own standard, ASTM F2459, that governs this particular test method. And with this gravimetric test, we use a particular solvent. And the benefit of this test is that we can use both nonpolar and polar solvents to capture water-soluble, non-water-soluble, and insoluble debris. And so this test has a wide range of the contaminants that it can detect. And so often our clients will choose multiple solvents, maybe say a polar and a non-polar solvent, to try and capture those all three types of contaminants can be captured within this test. And so we take the device and submerge it in water. We do a sonication step, which is a very vigorous shaking to shake off those surface residues. We remove the device. We evaporate off the remaining solvent, and then we're left with those surface residues, and that's what we weigh and quantify and report out with the final report. With this test method, it's also a requirement of the standard to determine the extraction efficiency. And what the extraction efficiency is it's a control with the test method to show that it's effective at removing the residues from your device. And the extraction efficiency can be determined two ways. The first being a spike recovery, where we take a clean sample that's gone through your process, we spike or dope it with a known amount of the contaminant, we do one extraction to get a percentage of the those surface residues that we were able to recover. And so that's one option to determine the extraction efficiency. The second method being the exhaustive extraction method. And with this method, we usually see an uncleaned or a dirty sample sent in where we do repetitive extractions until that final extraction is less than 10% of the original extraction or below our limit of detection. And with that exhaustive extraction, we're able to get a percentage of the extraction efficiency. And what that extraction efficiency shows is how effective the test method, and in this case solvent chosen, are at removing the residues from your devices. And that ASTM method requires that the extraction efficiency be greater than 75%. And so that's specific to the gravimetric test method to include per that standard. Another benefit of this gravimetric test method is that we can take the residues and scan them on the FTIR and attempt to ID components in the residue. And the FTIR, which stands for Fourier Transform Infrared Spectroscopy, where we take that infrared beam, scan those residues, and then we're able to get a spectrum of bonds or functional groups. And then we have a library of known compounds where we compare the residue to the library and try to get a match. And then we're able to report those results to our client based on what their matches are. For example, we were working with a client recently who was getting some high residue values with his gravimetric test, quite a few milligrams being left behind of residue. And we talked to them, said, hey, we could ID these for you to see maybe where this contamination is coming from. We scanned the residues on our FTIR, and we did a library search, and we got very high match numbers to oils. And so this client was able to go back to his process and implement a more rigorous rinsing step to try and remove that oil more effectively from his device and get his residues lower. And so this gravimetric test in combination with FTIR is a great pairing to quantify the residues. And then if you're getting a substantial amount of residues, we have this tool to help you maybe go back to the process and try to reduce the risk of that contaminant that's being left behind. The second test method that is an option to quantify those surface residues is TOC, or total organic carbon. And with this test method, we take the device and we do an extraction. And with TOC, we can only use water as the extraction solvent. And so this is a great test option if maybe you have water-soluble contaminants that are primarily comprised of carbon. And TOC is going to be a great option for those types of contaminants. So we submerge the device in water, again do a sonication or another rigorous shaking to remove those surface residues. We remove the device and take aliquots or samples of that extract and run it on the TOC instrument, and we're able to get a TOC, total organic carbon, readout of the oxidizable carbon that was present in that extract. So some benefits with TOC is that there's a very, very low limit of detection for this test method. We can go down in the part per billion range, and so TOC is very sensitive. However, also with TOC, it reports out a worst case value of the total amount of carbon that's present in the extract. So there's no specificity here, unlike kind of with the gravimetric test, we have the option of FTIR to try and ID or identify what's in that residue. With TOC, we give a worst-case quantitative amount of carbon, which also can be very beneficial. There's just no specificity here. And another benefit of TOC is that it's been around for a long time, and it's widely recognized in the industry as a marker for cleanliness. TOC is often, always included as well as part of reusable device cleaning validations. And so TOC is a great marker for cleanliness that's widely recognized. The third test method that we are going to discuss is VIS Spectroscopy, or ultraviolet visible spectroscopy. And with this test method, it works best with contaminants that readily absorb UV or visible light. And these are often detergents, and with this test method, we often see samples sent in to analyze how much detergent is being left over on the devices. And with this test method, each contaminant that we see undergo kind of a validation themselves before we do the testing. And we do this because every contaminant absorbs this light differently, and so every contaminant is going to have a different LOD and a different curve, basically, to where we are going to be examining how much residue is left behind. And so we have to make sure that each contaminant can scan well on the instrument and that we're able to validate it before we can, test if it's being left behind on the residues. And so with this test method, we also use water as the extraction solvent. So we submerge the device in water, again, do a sonication or shaking to remove those residues. The device is removed, and then that extract is scanned on the UV-Vis instrument. We also take a stock sample of detergent, scan it as well straight onto the instrument, and then we compare the two scans, the residue that was obtained from the extract to the stock solution of detergent. And then with that comparison, we're able to report a quantitative worst-case amount of detergent that's being left over on the device. And with these detergents, some detergents can have a very, very specific peak shape. And so we say, like TOC, that this test doesn't really have a lot of specificity and it's a worst-case amount of detergent left behind. However, if you're getting a very, very similar curve to your detergent and when it's compared to the residue, you can have some specificity there to show that, this is what I'm quantifying is actually the detergent. So this test option is a great one if you are specifically wanting to look at a detergent used in your process and how well that's being removed after you clean your device. And so there are some limitations with this test in terms of the contaminant has to be water soluble, and we have to be able to validate it on our instrument. However, it's very specific in looking at that detergent or that particular contaminant. And so this is a great test option, especially when you're trying to look at your detergent specifically. The next test option that we are going to discuss is the particulate test. And with this test method, we also do a device extraction, and then we run that extract through the liquid particle counter to get a quantitative amount of particulates left behind. And what we're talking about with particulates and this insoluble debris left over on the device, these are often maybe metal flakes or particulates, anything that you can see, and a lot of the times our clients know if they're concerned with some of these particulates or metal flakes or things of that nature. And sometimes people want to identify and quantify these separately. And so the test method for this particular test is USP 788. And there are limit values for injectables. And if you recall, when we were just discussing the gravimetric extractable residue test, we can also remove and quantify those insoluble residues with that test method. However, this particulate test method quantifies just the insolubles on their own. The gravimetric will quantify what's insoluble as well as what is soluble with the solvent that we used with that test method. And so you can also see as well with these 4 test methods that we've just discussed is that there's some overlap. For example, if you have a detergent that's water soluble, the gravimetric extractable residue test, TOC, if it was comprised of carbon, as well as the detergent residual test, could quantify that detergent. And so that's why it's so important to get that list of potential contaminants, and that's how you really decide what test method is going to be best for you and your process. And everyone is going to have a unique and individual plan based on what those potential contaminants are, as well as the material compatibility of your device. So there is some overlap here, again, with the particulates and insoluble debris. And so you just kind of have to look at what total you want to look at, and then decide which test method, either one or multiple test methods, are going to be best at quantifying those surface residues. And the next step in these cleaning validations, and probably one of the most difficult, is to justify the residue levels. And again, there's no regulatory limit or acceptance criteria for these cleaning validations as a whole, which makes it very difficult for manufacturers. And it's been difficult as well for regulatory industries and FDA to set a limit. There is such a wide variety of devices, the uses of the device, materials used in the process, and the different processes themselves that regulatory agencies have found it difficult to set a number. For example, how can you say that if you have 1 milligram, for example, of residue, and less than that you have a clean device? It's been very difficult to come up with one number. And so that's kind of why there's a lack of acceptance criteria guidelines in the industry for these cleaning validations. However, we do have some tips on what we've seen from other clients and what we've seen success in the industry for justifications that people use. First, we often see people compare clean and unclean devices. So they will take a dirty sample and test it, or an uncleaned, and then test clean samples. And then hopefully you see a reduction in residue there, and that can help show that your cleaning process is effective. And so these dirty to clean comparisons are often what we see to help show, that residue on the cleans is clean. We also see people comparing new versus old processes, and this is probably most prevalent when we're seeing people who have maybe implemented a change in their process like we discussed before, and they're trying to show equivalency between the two processes. And so these comparisons can help show, after you've established your criteria, if those changes were beneficial to implement or not. A lot of people will also use kind of historical data and trending or success on the market to set their limit values. A lot of people have had devices out on the market and have never had a problem, and maybe are going back and trying to have this validation data. And if they've had success on the market, they can maybe just start doing some routine monitoring, maybe set up some routine TOC tests, and then as they're consistently getting these results, if they're similar, set an acceptance criteria and a range based on what they're seeing. And so that's more for if your device has been out on the market for a while and you're going back trying to validate the process. We also see people compare the residue values from the extract to the analytical detection limits of the test methods. So again, these are the test methods for the analytical test that we just discussed. And some people will say if the extract, values are less than the limit of detection of the test, well, I have a pretty clean device. We caution people here, however, that we want to make sure that you are examining enough of the device in the first place to examine residues if they were present. So for example, if you have a smaller device that's on the smaller end, say maybe a smaller screw, and you test it and your results are all below the limit of detection, and so it looks like we were unable to detect any residue from your device, an auditor may question if there's really not a lot of residue there, or if we had enough weight or surface area in the first place to detect any residue if it was there. And so that's something to be cautious about with these test methods and using that as your justification for a clean device. And on a side note, in terms of sample size for these test methods, with the analytical test, there's no set sample size requirements for what you have to send. However, one of the tests we will be discussing next, the cytotoxicity or MEM elution test, that does have an acceptance criteria, and we recommend a similar or comparable amount to that cytotoxicity test to show, comparable results, which we will touch on later. And so that's a recommendation in terms of sample size for surface area or weight for these tests. Next, as a tool to help justify the residue levels, we often see people use LD50 value comparisons, where we see people, again, with that list of potential contaminants, maybe take their worst case or lowest LD50 value, and LD50 meaning the lethal dose to kill 50% of the population with that contaminant. And this data is often found on the MSDS sheet of the contaminant, for example. It's often a predetermined number that you won't have to do testing for. But a lot of people will take that LD50 value, put it into a calculation that makes it comparable to these test results, and then they'll say if the result from the extract is less than the LD50 value that you've calculated, I have a pretty clean device. And so we see that that's very common in the industry, that a lot of people are using these LD50 value comparisons, and so we did want to mention that in this presentation as a tool that you could possibly use if you feel that that would work for you in your process. And finally, to help justify the residue levels, we recommend pairing additional tests into the validation, such as cytotoxicity, endotoxin, and bioburden, to help justify those overall residue values. And with cytotoxicity, so this test has its own acceptance criteria. And again, this MEM elution test is a test used to show if the residues that are obtained with the test we just did to quantify the residues, this test will show if those residues are cytotoxic or not. And so this test, per USP 87 and ISO 10993-5, has its own acceptance criteria, where a passing test score here is cytotoxicity grades of a 0, 1, or a 2, and a score from this test. And so you can help justify that overall residue value with this test to show, I've got this amount of residue that I just quantified, but the residues are non-cytotoxic. That can help you say that you have a clean device. And so we always recommend including the cytotoxicity test included as part of the validation for this purpose. Also, you know, a lot of people will argue that they already had to do maybe cytotoxicity as part of their more comprehensive biocompatibility test plan that they had to do on their device. And that's fine. There's definitely no requirement that this test has to be done. It's just our recommendation to help justify that residue value to include it as part of the cleaning validation. To also examine microbiological contamination left behind, we recommend including endotoxin or bioburden data as part of the validation, again to help justify those overall residue values. And with this endotoxin or LAL test, again, endotoxin being a residue of gram-negative bacteria, it again has its own limits of acceptability from USP 161. And you can say, you know, if you include endotoxin testing as part of your validation, again, with that residue amount you quantified, maybe it also has passing endotoxin scores, that can help you say that you have a clean device. Same with bioburden. However, there's no set acceptance criteria for medical devices. However, low bioburden counts, maybe low counts of aerobic bacteria and fungi, for example, can again help justify that you have a clean device. And you can see with cytotoxicity, endotoxin, and bioburden, these are just tools to help and kind of, you know, give you more data to show in your overall cleaning validation that you have a clean device. And this endotoxin and bioburden tests are also mentioned in that overall governing ASTM standard that we mentioned previously, the 2847, that discusses these cleaning validations as a whole. The cytotoxicity test is not mentioned as in that standard, but that's just a Nelson Labs recommendation. And again, kind of like what we discussed with cytotoxicity, a lot of people may argue that their bioburden is going to have to be performed as part of their sterilization validation. And again, that's fine. There's definitely no requirement that says you have to include it as part of the validation, but you can see how it can be a helpful tool in providing more data about your device and trying to pass it off that it's clean. And so to kind of, we're nearing towards the end of this presentation, but I wanted to include maybe 2 case studies with some numbers of what we've been discussing today so you can kind of see better what we're talking about here. So in this first case study, this client was using TOC to help improve their cleaning process. They were using a detergent, and they wanted to see if maybe switching to a different detergent would leave behind less residue. And so with this test, they tested an unclean sample and got a result. And then they were currently using powdered Alkanox detergent in their cleaning validation. And that 161 part per billion is the result they got there. And then they were thinking of switching to liquid Alkanox or Liquinox detergent, which they were actually able to reduce down to 94 part per billion in their TOC results. So for this client and their device and their process, the Liquinox, it appears, was being, you know, rinsed more effectively from their device, and so this client actually ended up switching over to Liquinox instead of the powdered Alkanox to use in their process because it was leaving behind less residue. And that comparison between the unclean, you can see that drop there with both of the numbers from the 589 part per billion down. You can see that this cleaning process is actually doing a pretty good job of removing residues from the device. And this second case study that we have here is a gravimetric test with MEM elution and bioburden. And so what we have here is our client sent in a sample for an exhaustive extraction, so this was an unclean sample, and we did 3 repetitive extractions to gain the extraction efficiency, which was 91%. And so what that again means is that the test method and solvent chosen were able to remove 91% of the surface residues. So that's just a great control to include with the test method to show that it is working and we can detect the residues that are even there. And then they tested a clean sample with milligram amounts of 0.45. And you can see here that from that first uncleaned extraction from 6.45 milligrams, the reduction down to 0.45 milligrams. That's a great reduction and definitely shows that the cleaning process is being effective at removing residues from the device. And so, maybe with this test method there could be some more cleans samples tested for some more reproducibility. Say if they tested at least 3 cleans and got around that 0.45 residue, you could show reproducibility with the results. And so you could just see maybe having some multiple samples tested here can make that look even better with the results. Like, is that 0.45 milligram a fluke, or is that reproducible with the cleaning process? And then pairing this test with MEM elution, they sent in 3 samples and received all scores of 0, which show no cell lysis. And so they can also say with that 0.45 milligram residue that it's non-cytotoxic. And then this client also tested for bioburden, aerobic bacteria, and fungi, and they were able to receive non-detectable levels or non-detectable counts of this bioburden. And so they can also say with that 0.45 milligram value that bioburden is not a concern in this case. And you can see how these tests work together and how those other tests paired with the gravimetric can just help you in justifying that 0.45 milligram of residue and showing that you have a clean device. But again, also kind of what we talked about at the beginning, you can't just test for cytotoxicity or bioburden as part of the sterilization validation, because just because it's non-cytotoxic and it's sterile doesn't mean it's clean. And that's why it's really important to quantify as well how much is there and that it's a safe and acceptable amount. And so that's why these tests are important. And to list out some of the important references that we discussed today, the first is the GMP requirement that talks about, why you have to do these cleaning validations for manufacturers. And then the second and third references that are on this slide, the second ASTM 2459 is the test method for the gravimetric extractable residue test, that test having its own separate standard. And then the third and final standard listed on this slide, the 2847, is the overall cleaning standard that discusses these cleaning validations as a whole and the test options available. And thank you so much again for joining us today with this webinar. If you have any questions and would like to discuss any of the points in this presentation further, please feel free to contact me or Nelson Labs, and we would be happy to discuss your questions. Thanks again for joining us, and we hope to talk to you soon.